Welcome to the CCMB seminar. I have the pleasure of introducing Fernando Villanea, who is a postdoc in my lab, but he's moving on starting January. He got a faculty position in anthropology at CU Boulder, and I thought it was. I really wanted him to give, this is his exit seminar. So Fernando will still be collaborating with us. So it's not like a goodbye. But I wanted him to have an opportunity to give his last seminar as here at the CCMB slot. So he's going to be talking about some of the work that he's working on now. And it's been a pleasure to work with Fernando. I'm going to miss him. I think everyone's going to miss him in my lab. But I look forward to continue to collaborate with you and to work with you. And I'm really excited to see how your lab is going to grow. And so with that, I want to let you start.
Thank you, Amelia. Let me share my screen because I forgot to do that. Okay, can you see my slides? Yeah?
Yes.
Okay. Well, thanks, Amelia, for the welcome. I guess there's not much more to say except, yeah, I was a postdoc, or I am still at CCMB. I was very fortunate to be part of the Huerta Sanchez lab, where I had a great time, did a lot of good work, and also learned a lot. But I'm being kicked out of the nest, and so I'm starting my own lab now at the University of Colorado Boulder. I'm actually currently in Boulder right now, and it's a lab of one, but hopefully that won't be the case for long. And today I'm going to talk about the projects that I've been working on for the last 2 years, and it's a lot of them, so we're going to fly through most of them. But here's a quick overview of the 5 projects that I'm going to talk about today. And as you can see, the common theme is archaic admixture. It's this idea of sort of the legacy of Neanderthals and Denisovans and maybe other archaic species in humans today. And in general, I mean, this is a process that you can sort of generalize and think about it this way. So we have a human tree of life here with some human populations, maybe in the present down here, maybe some populations in Africa and some populations outside of Africa that connect back in time. And then if you go farther back in time, our entire human species connects with other species. And so we can think of this red lineage right here as a different species of human, archaic human, that went extinct at some point in time. But before they went extinct, they actually admixed with some modern humans, such that some of these modern humans carry some portions of their genome today. And if we look at this process from the point of view of just looking at genome sequences, we may see something like this, where we have different individuals, and some of them carry some portions in red of this archaic ancestry. And they don't have to be the same in all individuals, and we may find some individuals that just don't have any ancestry left. And some individuals may share some of that ancestry as well. So we find this pattern across human populations. And in general, there's 2 types of questions that I like to ask based on this data. The first one is, can we use this data to learn about the past? So can we learn something about this structure that I was talking about before, and maybe about this process, maybe when this happened, if it happened multiple times, what was the magnitude of these events? And the second types of questions are questions focused more on the present. So we can ask, are these archaic genome elements that we see in humans today, do they have any effect in human health or in fitness in general? And the reason this is a valid question is because if we look at this map, so this is a map of archaic genome elements in humans today, we can see that actually most of the world carries some amount of archaic DNA. And here is presented as a heat map. So cooler colors means less archaic ancestry, warmer colors means more. and we have ancestry for Neanderthals and also for Denisovans. And as we can see, most of the people today actually carry some amount of ancestry. This map is actually pretty old, and further sampling and refining of methods have shown that, in fact, people in Africa also carry archaic ancestry. In some cases from Neanderthal, but in other cases from even other species that we don't really know about. So this is something that we can find across the globe. But in general, just to catch you up on who these Neanderthals and these Denisovans were. So these are species that we've known about for some time based on the archaeological record. The earliest Neanderthal that were recognized as such, so recognized as a different species, have been known for over 100 years. This illustration right here is one of my favorites. It's from a newspaper from the London Times from 1909, in which at around that time, one of the Neanderthal skeletons became very popular in the news. But they were represented these sort of ape men, these cave dwellers, sort of really archaic, really primitive, maybe this offshoots of humanity that sort of were degenerated in some sense and were not really connected to modern humans. And since then, in the next 110 years, we've actually modernized that view of Neanderthals into thinking of them as very modern, basically just an alternative version of us, modern humans, where, and this is based on finding a lot more skeletons that are really good quality, lots of skulls, also tools and other cultural remains. But part of the reason we have this modern conception of Neanderthals is the fact that from some of those bones, we've actually been able to amplify DNA. And so we have the genomes of 3 Neanderthal individuals that are very high coverage. So they cover the entire genome. And there's actually more genomes that are partial. But from these individuals, we found that not only are Neanderthals genetically very similar to modern humans, we've also found that modern humans actually carry some Neanderthal elements today. And so because of that, calling Neanderthals primitive and ape-like ended up being sort of like a self-insult. And so we now just think of them as being sort of these archaic humans that existed in Europe and Siberia and were similar to us. As the story with Denisovans is very different. So as it turns out, from the same cave where one of the Neanderthal individuals that were genome came from, they kept finding some other subfossils. So they found this molar that it very much did not look human and very much looked like a Neanderthal molar, as well as some other pieces of bone, like this little pinky bone. And those look in really good, they look in a really good state, such that they could be sequenced. And so the expectation is that these were going to be Neanderthals. And as it turns out, the genome ended up being of a completely different species that is as different to Neanderthals as Neanderthals are to us. And so we call the species Denisovans. They don't have a species name because we don't have a holotype. We don't have a complete skeleton. We actually have very, very, very few fragments that are known for, but we do have a complete genome. And same as Neanderthals, we find some Denisovan ancestry in modern humans today. And so the story goes a little bit like this. So if we turn the clock back 400,000 years, we have our species, anatomically modern humans, evolving somewhere in Africa. It's not exactly clear where. And then we have Neanderthals roaming Europe and maybe venturing into parts of East Asia, like Siberia, where we also have Denisovans, which are thought to be an East Asian and Siberian species. But we also have these other species that we don't have genomes for, but we have bones and other archaic, some archaeological materials, like Homo ergaster in Africa, maybe more Denisovans farther south than we have evidence for, maybe Homo erectus, maybe Homo floresiensis in Southeast Asia and in Melanesia. And so there's a possibility that all these species may have also overlapped with each other, may have been exchanging genes. What we do know is that some 60,000 years ago, modern humans started to expand into the entirety of the African continent, and then maybe sometimes a little bit outside. And as they were expanding, they were encountering these other species, including Neanderthals in Europe. And they were actually admixing with these species. So they were incorporating some of their genetic material into the genome of our populations. And so that's represented in this little colored dot. Modern humans kept dispersing, and as they dispersed, they kept encountering different archaic human species, and they kept admixing such that they're carrying the legacy of multiple species. In by some 40,000 years ago, modern humans were the only species of human that were left. So all these other archaic species had gone extinct, and all that is left of them is what we carry in our genomes today. And the reason we study these and they're very interesting is because we have found evidence of some of these archaic genome elements to being really helpful for modern humans. One of the best known examples is the gene EPAS1, which is helped Tibetans survive in high altitude environments, which are also very low oxygen. And it turns out that this is a Denisovan variant of that gene that is found in most Tibetans at very, very high frequency. We also have evidence of some other genes that are related to the immune system. So they might have helped increase diversity of the immune system, as well as some genes that affect the skin color and hair color and might be linked with adapting to environments that have higher or lower UV light. And so I made this meme to represent this process. I made a lot of memes for this presentation. I hope that's okay. So in this meme, the small brain is this idea that, yes, humans inherited some genomic genome variants. The larger brain is this idea that some of these variants might have been adaptive in these archaic species, and therefore, as we gain them, we're actually getting a leg up by getting some of these genes that are beneficial. So we're kind of cheating the system that way. But the super brain is this idea that, you know, these archaic genome elements don't have to be specifically adaptive in archaic species. The simple fact that we're getting genetic diversity is giving us more of a substrate for natural selection to occur. And so we are way cheating the system by just incorporating all of this extra genetic diversity, and then that's allowing us to adapt to environments quicker because we have more genetic diversity to start with. And then the galaxy brain idea is also, when you think about other ways in which you can increase genetic diversity for natural selection to occur, such as processes such as mutation, they have a ton of drawbacks. So mutation is random. It can happen anywhere, including in perfectly functional genes, and it may disrupt genes. So these mutations may become deleterious. And so mutation is actually really, really dangerous. So it's a dangerous way to increase genetic diversity. But for these archaic genome variants, you know, they existed in the genomes of humans that were similar to us. So we're incorporating these variants that were kind of field tested in these other genomes. And so we're cheating by increasing the genetic diversity, but we're also doing it in a way that's very safe. We're taking these variants that are probably work to some degree in a human. And so just to jump into the projects that I've been working on, and the first one, these are not in chronological order. These are in the order that makes the most sense to me to, you know, just tell a story. I'm going to start with this review article that I've been working on with some other people also at CCMB. So this kind of started as our journal club. We had a journal club where we were reading a lot of papers on archaic admixture and tools to identify archaic admixture. And as COVID hit, we couldn't meet in person anymore. And so Kaylee had this idea of, hey, why don't we keep meeting online? And since we're reading all these papers anyway, why don't we write a review paper? And since then, I'm being really proud to see how this paper has kind of come together into this really, really cool review article that is probably going to go for publication pretty soon. And I've been really lucky to work with all these different people that have collaborated. We've collaborated together. But for the specific portion that I was working on this idea of reviewing the literature, looking for every instance in which a study has found evidence of introgression between an archaic human and modern humans. And so in this figure, this is present time. This blue lineage are modern humans in Africa, in Europe, in Asia, in Melanesia. And these are connected back in time. So time goes back this way. And then we have these other archaic species. We have Neanderthals in red, which are both of this European types and the Siberian types. We have Denisovans in Siberia, and we have Denisovans in the Oceanian parts of the world. And we even have this yellow line, which we call the super archaic species. So these are species that are even farther diverged from humans than Neanderthals and Denisovans. And we don't have genomes for them, but we do find some evidence of admixture in the past. And so each of these dotted lines is a case of a study that's pinpointed a specific admixture event between all these different species. And the take-home message that I kind of want to point out here is that there is a ton of them. So and all these studies are very new. This is in the last 5 years, and most of them are actually from last year, from this year. So in the last couple of years, we've kind of coalesced into this idea that there's been a ton of archaic admixture. There's every time in which these archaic species have encountered humans, they have interbred and they have interbred with each other, which has led me to make this second meme. This idea that based on how many of these introgression events are there, are we actually archaic humans? And the answer is yes, we always have been. We have been carrying all of these archaic genome elements through most of the history of our own species. And so we're looking forward to submit that review article, but I'm going to jump into a different project, which is looking at ABO blood group systems in Neanderthals and Denisovans. This is a project in which Amelia and I have been collaborated with Dr. Keola Fox at UCSD, in which we are sort of looking at the ABO gene in Neanderthals and Denisovans and just asking, what do they have the same A, B, and O variants that humans have? We are imputing genotypes from these archaic genomes, and we're also calculating genetic distances so we can identify if some of these ABO types in these archaic species exist in humans today. And this paper is actually in review right now. It's about to go. I mean, we're about to send revisions and hope this is the last one in review, and we actually get to see it published pretty soon in MBE. But in sort of the general concept of why it's interesting to study ABO, because ABO is being this classic marker. It's one of the earliest genes for which we have evidence of genetic diversity, for which we don't even need to sequence, because we can do serological tests that can tell us a little bit about human variation. It's also really important for blood transfusions and organ transfusions. Basically, if you have a blood transfusion from a donor to a recipient that is of different blood types, the recipient may actually have an immune response and may die because of the immune response. So it's literally studied from the medical perspective, such that we know which functional mutations actually lead to each of the different phenotypes. And because we know which mutations lead to which phenotypes, we can actually impute phenotype data just by looking at sequences. And so that's what Keola Fox did. So he took the 1,000 genome panel sequences, and then he just turned each of those into a phenotype for the A, B, and O types. And he even found some of the rare types like A2 and AB. And so this is the 1,000 genomes data in blood types. And to do that, he came up with this table that has 68 variable positions that actually have a link to a phenotypic ABO variant. And because he had this table, it was very easy for me to then just look at these positions in these archaic genomes and identify which functional blood types they had. And so I asked 3 questions. The first one is, what are the ABO alleles that are found in these archaic genomes? I then asked, are any of these found in modern humans today? And then finally, I asked, is this the effect of balance in selection? And I will get into each of these 3. So to recap really quickly, these are the results. So we found that this is the Altai Neanderthal, which is found in Siberia, and it's the oldest. The Chagiskaya is another Neanderthal from Siberia. There's a single Denisovan genome that is also found from Siberia. These are all found from the same region. And then there's the one European Neanderthal, the Villanea Neanderthal, which is found in Europe. And we find that the Altai Neanderthal actually is a homozygous for this O2 allele that is also found in the Chagiskaya Neanderthal. The Chagiskaya is a heterozygous for that type in an O1 type that is also found in the Villanea Neanderthal in Europe. So there's a connection between the Siberian and the European Neanderthals. And we find that both this Altai O2 and this Villanea O1 are found in modern humans today as well, in East Asia, in Europe, and in Southeast Asia. We also find that the Denisovan is a heterozygous for 2 different types of O, an O1, and a really rare O with a B background. And we find that the Villanea is a heterozygous as well for another rare type, which is a B with an A background. And so we found what alleles existed in these archaic individuals, and now we're asking, are any of them shared with modern humans? And so for that, we just calculated genetic distances, which we plotted here. So this is the Denisovan individual, for example. Genetic distance is represented on the y-axis. So closer to the bottom is more closely related to Denisovan. Farther away means is more different from Denisovan. These are the 3 Neanderthals. And each of these is a global population from the 1,000 genomes panel. So we have Europeans in blue, we have Southeast Asians in yellow, we have East Asians in red, and we have African populations in green. And this dotted line is sort of the level at which you find the average for both of the Denisovan haplotypes. And as you can see, and each of these dots is a chromosome. So it's half of a person. And as you can see, a lot of these chromosomes are pretty close to Denisovan, but they're not quite there. And in particular, in Africa, we see some of these chromosomes that are very close to Denisovans, but then they're not quite there, but they're close. And we interpret this to mean that the Denisovans carry types that have been maintained in the human Denisovan lineage and are found in peoples in the world today. But they've changed a little bit since then, since they're not directly related. And it makes the most sense that in Africa, we find the closest types because Africans have the highest genetic diversity. But this is no evidence of introgression. Whereas if we compare that with the Altai Neanderthal O2 type, which is right here, we first find that it's different from the other Neanderthals, which is interesting. We find that most modern human chromosomes are very different from the Altai type. But then we find these outliers right here in Europe and in Southeast Asia. And these are so different from these other chromosomes that we find up here that they actually present a very good evidence for introgression. We think that these are actually Neanderthal types that survive in modern humans because of admixture with Neanderthals in the past. If we look at the Villanea Neanderthal, we find a different but similar pattern in which the Villanea and the Chagiscraia are very closely related, and they're different from the Altai and the Denisovan. And then we see most humans do not have a Neanderthal type, but we find in East Asia, a couple of the populations have this Villanea O type. And we again think this is introgression from Neanderthals. The Chagiscraia Neanderthal is very similar to the Villanea. Same pattern, different from Altai, similar to Villanea. Not really found in Europe or Southeast Asia, but found in a few individuals in East Asia. And so to sort of recapture, this is the pattern that we see, where we have the Villanea Neanderthal and the Chagiscraia Neanderthal in blue. And then we found very few of those chromosomes in East Asians today, so very, very low frequency. And then the Altai Neanderthals in red, and we found those O types in Europe and Southeast Asia, again, at very low frequencies. And what's interesting is that there's this sort of inverse geographic pattern in which the Altai Neanderthal is Siberian, but we find that type in Europe, and we find that type in Southeast Asia, but we don't find it in East Asia. And then we have this East Asian type that matches the Chagiscraia, but it also matches the Villanea, which is an European Neanderthal. And at least that part kind of makes sense. So this is the results from a study that looked at the Chagiscraia genome, in which here is Chagiscraia, and this is basically like how closely are all these European Neanderthals to Chagiscraia or to the Altai Neanderthal, which people call Denisova 5 based on the archaeological sample. And what we see is that for all these European Neanderthals for which we have partial genomes, including the Villanea, for which we have a complete genome, they're very closely related to the Chagiscraia, which is the Siberian Neanderthal. So it kind of makes sense in that context that even though we have a Siberian and a European Neanderthals, there's a connection between the O type that we find that we're also finding in modern humans in East Asia. Now, why we find them all over the place? It's kind of like a shrug emoji. We don't know. It's very likely that Neanderthals have really complex geographic dynamics where some of their populations that have been moving all the time might have been replacing other populations and then being replaced back by the original inhabitants. So there's this very complex pattern that we don't quite understand, but we have a lot of hints. And so now we know that there are, in fact, some archaic alleles that are found in modern humans. And then finally, real quick, we wanted to see if this were the effect of Bayesian selection. The reason is because archaic ancestry in genes tends to be really bimodal. So you either find very few genes like EPS1, which have been helpful in modern humans, and you find those at really high frequencies. But then the vast majority of genes in humans don't have any archaic ancestry. And if you look at Neanderthal and Denisovan ancestry in human chromosomes, most of them are these gray areas. And these are called introgression deserts. So these are areas that are completely depleted from archaic versions, and they tend to be really gene rich. And so we think that negative selection over time actually remove a lot of these archaic variants that just were not working in modern humans. In some estimates based on, so this is an estimate of the trajectory of archaic ancestry, starting from 10% might have become as low as 2% because of negative selection in as few as 10 generations. That is extremely strong negative selection, removing all these archaic variants. And so the question is, okay, we find these Neanderthal ABO types, were they under which kind of selection? And as it turns out, we know that the ABO gene is under Bayesian selection, which is a really rare, really unique type of selection in which variants are maintained at as highest diversity as possible. So in ABO, we have the A, B, and O alleles, and they're always maintained at this equilibrium frequencies in which the O is twice as common as the A and the B. And we can have populations that start with frequencies that are really outside of that equilibrium, and really quickly selection will bring them back into that equilibrium. So we know that these ABO types are retained over very long periods of time because of Bayesian selection. And so the question is, okay, we see all this Neanderthal polymorphism. Is this evidence of Bayesian selection? And so to kind of get at that question, which is actually pretty difficult to study, we took advantage of the fact that we have the complete genomes of the Villanea Neanderthal and the Altai Neanderthals. And so these are individuals that were part of a population that were actually not the same population that interbred with modern humans. So in this phylogeny here, we have modern humans, we have Neanderthals, and they are substructured into the Altai, which is very divergent, the Villanea, and we have this population that I call Neanderthal X, and that's the actual population that interbred with modern humans. And so we can take all the surviving genome fragments in modern humans, and we can plot them based on the Villanea and the Neanderthals. And then we can see where that lands. And so the expectation is that a lot of these genome fragments are going to be right here, right? They're going to have a high match to the Villanea and a high match to the Altai. They're sort of like generic Neanderthal fragments, sort of the genome fragments that all Neanderthals have. And then we're going to see some other stuff that is going to be all over the place. And so when we plot all these surviving genome fragments from Europeans and from East Asians, this is the pattern that we see. So as expected, most of those fragments are over here. These are sort of like these general, generic Neanderthal genome fragments. We see that more of them match the Villanea Neanderthal than the Altai, and that makes sense because the Villanea is more closely related to this Neanderthal X population. And then we see over here, there's some genome fragments that don't match to either one of them. And as it turns out, those are probably the Denisovan genome fragments. So they're archaic, but they're not Neanderthals. They don't match to either one of them. Another question is, when I highlight where the fragment that contains ABO is, where is that going to fall? And as it turns out, it falls either outside of all the other fragments or on the very edges of all the other fragments. And so we take this to mean that as opposed to all these fragments, which have a common evolutionary history, probably share a lot of genetic drift, these ABO types are actually divergent. They're actually being maintained in the population at a higher genetic divergence than all these other genome fragments that are evolving neutrally. And so we think that is evidence of Bayesian selection. And so to conclude, yes, we find all these ABO variants in these archaic humans. Some of them are really ancestral, like the Denisovan versions. Some of them are really derived, like the Neanderthal versions. But we also find that these derived Neanderthal versions are found in modern humans today, in Europe, in Southeast Asia, and in East Asia. And we also think that because the genetic diversity of these fragments is really high, that in fact they were maintained through Bayesian selection. And so now I'm going to jump into two other projects that I've been working on. These are more recent projects. The first one is looking at genome coverage in Neanderthals. So this is a project that I've been working with Kelsey Witt in Emilia as well. It's part of this sort of larger project. And what we're asking is, can we use genome coverage as a source of data to understand the shape of the events of a human and Neanderthal contact? Part of the reason we're asking this question is because there's this idea that East Asians are enriched for Neanderthal ancestry relative to Europeans. And so we set out to test this using computer simulations. And eventually these are going to be folded into this sort of larger paper where we're looking at all these measures of archaic ancestry. But to recap, so we have these two maps. The map on the left is the distribution of Neanderthals in Europe and Siberia based on fossil sites. And as you can see, they're heavily based in Europe. There's actually very little evidence of Neanderthals in East Asia. But if we look at the map that I showed you before, the map of ancestry across the world, where cooler colors are less ancestry and warmer colors are more ancestry, we can see that Neanderthals or Europeans actually have a lower fraction of Neanderthal ancestry than do all other populations in the world. And that is not at all what we expected based on what we see in the fossil record. And so there's sort of this mismatch distribution, and it's sort of being the source of a lot of debate as to why this is. And so it's been approached through a number of different methods, but the way we're approaching it right now, which is novel, is looking at genome coverage. And so genome coverage is looking at when you have these genome fragments, where red here are Neanderthal fragments in a blue human genome across different populations. It's just looking at the distribution of those fragments. And so we can imagine that we have some European genomes, and those European genomes have some Neanderthal fragments in red. We also have East Asian genomes, which also have some Neanderthal fragments in red, and some of them are shared and some of them are unique. We also have African genomes, which tend to not have any Neanderthal ancestry. And then we have the actual Neanderthal genomes, which are all red because they're all Neanderthal ancestry. And so previous studies have looked more at the frequency of Neanderthal variants across populations. So here we're just counting sort of the overall number of these Neanderthal fragments in a population. And in this case, as we can see, there's only 9 in Europeans, and there's 11 in East Asians. And so we can say that East Asians are enriched for Neanderthal ancestry when we're looking at the frequency. So this is what we look at. This is what the data looks like when we look at the frequency. But the way we are computing genome coverage is by only looking at the unique fragments. So we don't care how many times they're repeated. We care of how much of the Neanderthal genome we can recapture based on the population data for Europe and East Asia. And as we can see in the very same example, we have 7 unique fragments in Europe and 5 unique fragments in East Asia, which means that in this case, Europeans are actually enriched. You can recapture more of the Neanderthal genome in Europe than you do in East Asia. And so when we plot the actual empirical data, this is what we find, where in this figure, so I'm going to walk you through it because it's a little bit complex. But what I've done is I've summarized genome coverage based as a ratio of coverage in East Asia over Europe. So this line, the red line right here over the 1, is the perfect ratio match. So if they have the same amount of gene coverage, they're going to be at the 1. If a pair is above the red line, there is more coverage in East Asia. If it's below, there's more coverage in Europe. And then over the x-axis, this is my sampling. So in this case, what I'm doing in the most extreme case is I'm only sampling a single individual in East Asia and a single individual in Europe. I'm comparing the ratio of genome coverage, and then I'm plotting them right here. And then I'm doing this a bunch of different times such that I can get a distribution. And so walking over the y-axis, what I'm doing then is I'm just increasing my sampling. So I'm grabbing 5 individuals randomly at a time, 5 individuals from the other population. I'm looking at the ratios and I'm plotting them right here, and I'm just repeating a bunch of times. And then I'm doing that across all different samplings up until I get to really high samplings that they match the number of individuals in the 1,000 genomes panel. And so what we find is that in the extreme example where I'm only looking at single individual pairings, we find that East Asians actually have a higher frequency of Neanderthal ancestry. And there's a lot of variance, but in general, they sit kind of around here at 1.2, so 20% exceeding what you see in Europe. And that is actually a match to what you see when you just look at frequency data. So this is the enrichment that is classically looked at um, in archaic ancestry studies. But as I increase the sampling, we start to see that kind of advantage in East Asia starts to get lost. And as that sampling gets pretty big, in fact, it's the Europeans that can recover more of the Neanderthal genome than East Asians. And so we call this is kind of like a smirk pattern. And so the questions we're asking is, okay, why do we see this? So what are the demographic forces that are contributing to this kind of unique pattern, which is very different if you look at frequency, and it's very different if you look at genome coverage. And so there's a number of different hypotheses that could explain this. The first one and most basic one is looking at the demographic differences between East Asian and Europeans. We know that through the recent history, East Asian, the original population that the founding population for East Asia went through a very stringent bottleneck that was more stringent than the bottleneck you see in Europeans. And they also recovered quicker. So they went through a really fast recovery. And it's possible that that little bit of a difference in the sense of the bottlenecks might actually be enough to produce this pattern. So in this case, we would have people in East Asia, and they have all these Neanderthal elements in red and also now in green, just to highlight them. And then they go to this bottleneck and recovery, and the green fragments are lost because of the bottleneck. But then because of the recovery is really quick, the red fragments actually become overrepresented. So they're overshared between all these different individuals. The second hypothesis is this idea that there could have been multiple episodes of interbreeding, and there's something that I've studied in detail in a previous publication, in which we find that both in Europe and East Asia, there is evidence for multiple pulses of Neanderthal introgression. So we think there's been individual events at all these populations, and they each have produced a little bit of extra Neanderthal ancestry in both of them. And it's possible that because these are independent and they're of different magnitudes, they might produce this pattern that we see. And so here the idea is that we have these East Asian genomes, and then there is the red is Neanderthal ancestry that is also shared with Europeans. But then you have secondary integration that sort of adds some of these more rare fragments. And these are also added in Europe, but they're independent. So maybe there's more of them in Europe. The third hypothesis is this idea of dilution. So this was presented in this paper from 2016, where if you look at the ancestry of Europeans, modern Europeans come from this composite population that is made of all these different groups that have migrated into Europe at different times. And so there's this idea that there were hunter-gatherers in Europe, and then they met these pastoralists, and they sort of blended together, and then these farmers came in, they blended together. And so they have this really complex ancestry that is represented here in this illustration. And it was possible that one of these populations that blended into Europeans might have not have any Neanderthal ancestry. And so in this paper, originally they called them basal Eurasians, which is not a real population that exists. But most likely these were actually African populations. These are African individuals that are continuously poured out of Africa, and they've made it all the way to Europe, and they've blended with the Europeans. And so because they never interbred with Neanderthals, they have lesser, so they overall diminish the Neanderthal ancestry. So in this case, we have, again, these red and green Neanderthal genome fragments, and then this dilution, which we add individuals that are completely blue, so they don't have any of these fragments. And so when you consider them as a population, they actually lose archaic ancestry in terms of frequency. And so to explore which of these, if any of these, explain that smirk pattern that we see, I built this complex demographic model in which I can simulate all sorts of things, including multiple pulses of introgression. I can simulate the different bottlenecks that we see in Europe and East Asia. I can also simulate this dilution by taking some of these Africans and putting them into Europe. And so then looking at the simulations, I can then recapture that the graph that has the smirk and see if we find it. And so looking at the actual results, so this is what happens when you only have one pulse of introgression. And then this is only looking at demographic differences. So this is only looking at those different bottlenecks between Europe and East Asia. And as we can see, we don't actually see that symmetric pattern, but we do see that having different bottlenecks does influence the distribution of these ratios. So it actually makes it so Europeans have more genome coverage at the sampling level of 1, which is the opposite of what we see. But it does have an effect when we simulate two different pulses of introgression, in this case, a big pulse and a really small pulse. We see something similar to the symmetric pattern. We actually, like, having additional introgression into East Asia makes it so at the individual level, you see more Neanderthal ancestry, but that advantage gets nullified once you go back to the population level. So this is very similar to what we see before. If we exaggerate that second contribution, so the second pulse of integration, and make it really large, we actually see an exaggeration of that pattern where now at all levels, East Asians have more coverage than Europeans. So it's different from what we see in the empirical data. And then finally, if we have the dilution, so where Europeans are receiving all this influx of individuals from Africa that don't have any Neanderthal ancestry, we see once again that we recapture that smirk pattern. And it doesn't dip as low, so it's not quite a match, but it's very similar. What's weird is, like, at this point, mixing integration and dilution hasn't produced that same pattern. So we haven't quite pinpointed the exact combination of multiple pulses of integration and dilution that can actually produce the pattern that we see in the empirical data, but we're getting really close. And so to conclude this portion, I need more simulations. I need to do more. But we do see that the bottlenecks affect the sharing of the rare variants. We see that secondary gene flow also reinserts some of these rare fragments into East Asians. We see that dilution lowers the abundance of rare fragments in Europeans. So we're getting there, and we just need to simulate more. And to finish us off, because we're running out of time, I'm going to talk about this next project is looking at this Denisovan MUC19 in American populations. So Kelsey, Emilia and I are working on this project where we're looking at this gene called MUC19, where RP, which is an undergrad in our lab, has also done a lot of the work. And we're asking the question, is this MUC19 gene introgressive in humans? If it is introgressive in humans, do we find it in indigenous American populations? And if we do find it in indigenous American populations, is it found at above average frequencies? And if that's the case, is this because of positive selection? And the reason we think that might be the case is because indigenous Americans actually have a really unique and really cool evolutionary history. So if you remember where we left our little vignette of human history, up to 80,000 years ago, we still don't have really humans in the American continent, but we do have people in Siberia. And the people in Siberia actually carry Neanderthal and Denisovan ancestry. And so 80,000 years ago, the first people started to migrate into America through the Bering Strait. In those 18,000 years, they actually end up moving through the entire American continent, carrying these archaic ancestry components. As they move into the continent, they're running into all these different environmental niches. So remember, these are all sort of circumarctic peoples in Siberia. But then as they're moving south, they're running into pastures and deserts in Caribbean islands, in high altitude forests and rainforests, and just sort of high altitude environments with no forests, and also even some even harsher circumarctic environments. So running into all these different environments, and that's just, you know, that's just the engine that drives natural selection. But they're also on their own, did all these really cool things. They domesticated hundreds of species. They invented agriculture. They developed these complex societies. They invented thousands of languages. They invented math and calendars and astronomy, and they built these giant monuments. So they were doing a whole lot. They were overcoming these environmental challenges, and they were thriving. And so we think that one of the factors that allowed them to thrive in all these environments is having Neanderthal and Denisovan ancestry, and that they might have co-opted that for positive selection into finding all these advantageous, using all these advantageous variants to adapt to their environments. And as it turns out, in a previous study where they were looking at candidates for positive selection in indigenous Americans that came out last year, they had a list of candidates, which the top candidate was this gene called MEC19. So this is the top candidate for positive selection that is unique to American individuals. But as it turns out, in a previous study from 2016, coming from Emilia's group, they had actually detected MEC19 as being an introgress gene for which the denisovan version is found at really high frequencies in American populations. And so it's possible that the denisovan version of MEC19 is actually what's driving the signal of positive selection. And so once again, we calculated genetic distances to find out that that's the case. So this is the denisovan right here. Distances are closer to the denisovan at the bottom and more different at the top. These are chromosomes from individuals in the 1,000 genomes panel from different populations. So Africa is right here in purple. Europeans are in blue. Southeast Asians are in green. East Asians are in orange. And then American individuals are in red. And we, as we can see, we have haploids that are sort of all over the place. So we have some of them are really different from the denisovan version, as expected, and most of them are in African populations. We find some denisovan introgress types in Africa, and those are actually found in the 2 samples that are taken from African individuals of African ancestry in the Americas. So this is the American Southwest, and this is the Caribbean. And so we think that those are actually admixed individuals that have some indigenous American ancestry as well. But we do find the denisovan version in all populations. So we find them in Europeans, we find it in Southeast Asians, we find it in East Asians, and we find it in Americans. And so now we're asking, okay, so they're found everywhere, but is it possible that American individuals have them at high frequencies? And so we here on this panel are looking at sort of the range of frequencies between the different populations. And as we can see, it's somewhat low in Europe. It ranges from 0.5% to 3%, a little bit higher in Southeast Asia, 5% to 12%, in East Asia, 2% to 11%. And in America, we find it at frequencies between 5% to up to 30%. So there's some indication that this denisovan version is more represented in these American individuals. The thing is that American individuals, like myself, we're admixed. So we have an American part of our genes, but we also have an European part of our genomes that comes from the European colony. And we also have an African component of our genomes that comes from the African slave trade. And so it turns out that it's possible to separate your genome components based on your ancestry. And so that's what we did for these American populations, where here, this is the African component of Colombians, this is the European component of Colombians, and this is the Native American component of Colombians. And the same for Mexico in yellow, Peru in green, and Puerto Rico in blue. And what we find is that then if we only look at the frequency of the denisovan haplotype in the Native American portion of their genomes, we find it completely overrepresented, from as low as 14% in Colombia to as high as almost 50% in Mexico. So almost 50%, half of the individuals carry this denisovan variant. This is extremely high frequency, very different from anything else we see in the world. And so we have some good evidence that there could be positive selection driving that frequency, and that MEC19 might be doing something different and special for American populations as they were adapting to their unique environments. The thing is, we don't really know what that is. MEC19, I mean, we know that it's a gene that controls this gel-forming mucin, and it's expressed in lacrimal glands. So it's related to your tear ducts, to crying, and also making snot and making all sorts of other body fluids that are viscous. We know from this paper from 2008 that MEC19 is also associated with this genetic disease called Jordan syndrome. So there's some relation to this deficiency of just MEC19 not working properly. And we know from this paper from 2018 that indigenous Americans actually have a really high incidence of Jordan syndrome. So we think there's some connection, but it doesn't make a whole lot of evolutionary sense. And at this point, it's another shrug emoji. We still don't quite know what the link is there for MEC19, but we do find it overrepresented in American populations. And so to conclude, we do find the Denisovan haplotype of MEC19 in all these non-African populations, but it's at much higher frequency in indigenous American populations. And now we're following up, we're doing these branch length specific tests for positive selection. So we're gonna rule out that this is random and that it's in fact positive selection, and that MEC19 is very different from other genes. And the last thing that we're going to do is to look at MEC19 in ancient American individuals. So these are individuals that are pre-colony, and therefore they only have the Native American portion of their ancestries. And that links it to my last project, which is a grant that I wrote when I started working in the Media Lab, and we were awarded that grant to look at archaic ancestry in indigenous Americans. And this is a collaboration with Dr. Maria Avila Arcos at the Autonomous University of Mexico, and Dr. Villanea Villa as well. I'm also collaborating with David Petty, who's a grad student in the Media Lab. And we basically took the funding from Leaky and we used it to generate these ancient genomes from Mexico. I also curated all these publicly available ancient genomes of good quality, so anything above 1x coverage. There's not that many. There's about 20 in current literature. And then we had 8 different individuals that may have yielded DNA. They were good enough for DNA. And we used that money to get 3 high coverage genomes from those individuals. So only 3 of them right now are high coverage. But it's actually, it actually pretty much increases my sampling by 15%. So I'm very happy with the way we use those funds. And we actually haven't really gotten any farther on that, but we're going to be looking at that in the future. I'm going to be carrying this project into my new lab probably and working on this for a long time. But I'm really excited just to see what we can get out of these ancient genomes. And so some general conclusions from all these projects. The first one is that Emilia is overrepresented in all of them. We meet a lot. I see Emilia all the time. But also the overall conclusion is that, like with all things, the real wealth is been in all the collaborations that I've made. I've gotten to work with really wonderful people, and I'm really excited to see where all these projects go and how we are going to keep collaborating in the future. And so just finally, I want to say thank you to everyone at CCMB, people in the Huerta Sanchez lab, people in the Ramachandran lab, to Meg, to Alden, Nathaniel, specifically Nathaniel for keeping us fed during all of those journal club meetings, for providing us with pizza. I also want to thank my collaborators in Mexico and in San Diego and the Leakey Foundation. Thank you.
Thank you for sharing your work with us, Fernando. It's exciting to see the kind of the big picture of all the stuff you've done at CCMB. Does anyone have any questions?
I have a question.
Oh, hey, David.
Hey, Fernando. Cool stuff. So I'm going to follow up on this notion of the dilution hypothesis in Europe.
Mm-hmm.
So there was this paper from David Reich groups, right, on the decline of the Paleolithic in these ancient so-called modern humans in Europe in the proportion of Neanderthal ancestry in those genomes over time. So from 50,000 years ago to 10,000 years ago, the Neanderthal segments decrease rather linearly. And then there was a paper followed up on that from, I think, Svante Pabo's group or something, saying, sorry, pal, you got it wrong,
mm-hmm.
because it's not just due to progressive selection against deleterious Neanderthal introgression segments, it's a dilution due to back migration from either these putative ancient Europeans or maybe Africans or whatever. So, so what's the consensus on the purifying selection against Neanderthal haplotype? So in that, I think in that Svante Pabo lab group paper, they also showed that there was deleterious evidence for some purifying selection against certain areas, but it was localized to certain parts of the genome. So it's totally, you get these high profile Nature papers saying this is what's happening. And then you get another high profile PNAS paper saying, no, something else is happening. So what's happening?
Yeah, I mean, so, it's just been a really fast evolving topic. And so, yeah, this 2016 paper, they claim, okay, we're seeing this decline and we don't know why. So we're going to invoke this ghost population, basal Eurasians. We've never sampled them. They don't exist today, but it might explain this pattern. And at that time, they were not really considering gene flow from Africa, right? And so, yeah, this 2019 paper, which actually had a slide for that, but then I cut it because I was already probably going to run out of time. Yeah, they're saying no. In fact, we find that the decrease due to negative selection was very steep. So really quickly, you lose most of the Neanderthal ancestry, and then whatever you have left has remained the same over time. So instead of being this slow, removal, it's just a very steep removal followed by sort of this flat line of ancestry. And it's been revised a number of times. For example, I think right now there's some indication that these deserts of archaic ancestry have less to do with the fact that they are rich in genes and more to do with the fact that they recombine really slowly. So it could be that you see these deserts in portions of the genome recombine slowly. And so what's happening is, yes, there's very strong negative selection really quickly, and it's just dragging entire portions of chromosomes. But it's a fast moving field. I mean, I don't think there's quite been a consensus. I think right now, and the reason I'm modeling it right now as having this continuous African pouring of people into Europe is because it kind of makes the most sense based on the pattern that we see. I think your sound cut out.
In the David Reich paper, the synonymous, non-synonymous, or the deleterious and benign versus, significant amino acid changes also look like there was more deleterious load in the Neanderthal. So the argument that the Neanderthal genome, and then Miller and Rasmus had a paper on strong selection against that the Neanderthals were low fit populations. So is that confounded by this demographic migration story, or is that pretty solid? The notion that Neanderthals had a low population size and carried a lot of deleterious load.
So it's true that most. So it's an interesting question because the population of Neanderthals that interbred with modern humans were probably alive like maybe 100,000 years ago. But then we have these, and that's what we call late age Neanderthals, right? And then we have the complete genomes of the Villanea, which is only 50,000 years old, which makes it a very, very late Neanderthal. And the Chinese cry is 80,000, and then Alta is about 120,000. And we see this very long runs of homozygosity, which are, yes, indicative that they were in fact had very small effective population sizes. They were pretty inbred, and they have therefore a pretty big genetic loads. And it's a little bit difficult to say because we also know right now that there's a lot of substructuring, but also a lot of replacement. And so it's kind of difficult to know if that was all Neanderthals, or if that happened to be just specific, pockets of them. But the other thing that's interesting is that regardless, because they have a much more effective population size than modern humans, selection is just going to be much more efficient in modern humans. And so it could be that these weren't exactly strongly deleterious versions in Neanderthals. It just happens to be that when you put it in a human population, then all of a sudden natural selection is supercharged. And so
right. Okay, thanks.
Wish I had a better answer.
Yeah, well, I'm glad to know that there. Yeah. that you're as confused as I am. So good.
Right.
Yeah.
Thanks for the question.
I had a question.
Yes.
Yeah, it was a great talk, Fernando. I had a question about the American indigenous part, which I find really interesting because, as far as we know, unlike Eurasia, archaic populations never made it to America. And so all the archaic ancestry in America is the product of these like serial bottlenecks down the trajectory from north to south. So to what extent does that history of bottleneck demography affects our conclusions about about positive selection based off of these like high frequency archaic haplotypes? Because you can imagine that all those bottlenecks might also produce haplotypes at high frequency just due to chance. Hello?
I know he said he had kind of spotty internet connection, so it's possible that he's disconnected. So we'll hold on a second and see if he comes back. New apartment problems.
I had oh, sorry, am I interrupting? I had a very quick question, which is I was wondering if in the simulations, if you add.
I think he's gone.
Fernando is actually disconnected, so he may come back in a minute. I'm sure he'll be happy to take questions then, but.
Okay, sorry.
No, it's fine. He said he had his phone handy, so. Welcome back. You're muted.
Sorry about that. My internet is not great right now. Okay, sorry. I don't know how much of that you got.
I think it paused right before you answered.
Again. Oh, okay. It was a great question. Definitely have to consider strong genetic drift when looking at anything in Indigenous and American individuals. And the expectation across the genome is that archaic variants, because they're rare, they actually should be removed from the genome. But it's just as likely that any one of those fragments might just go, can go as high as fixation just because of random demographic forces. And so that's something that we have to take into consideration. So that's the reason we're doing this branch-specific tests, to see if this specific fragment is different than what we see in other genes that are also introgressed. But I think, so for future analysis, I think what we need to do is we need to model exactly like, what is the range of, what is the most extreme range of the high frequency that we should expect in Indigenous Americans given these bottlenecks. I mean, these bottlenecks are not exactly, all absorbing. Like Indigenous Americans still have somewhat high genetic diversity, just very low compared to all other populations. So yeah, it's something we have to look at really carefully.
Thank you.
I think Catherine had a question as well.
Yes, and I want to really apologize for jumping in accidentally. Our Christmas tree was arriving, so there was a moment of confusion, and I didn't mean to jump the queue, so I'm sorry. And my question is about the simulations, and I was wondering, it's just a quick question. I was wondering if there are more than 2 instances of introgression. Do you see a better match with the empirical data?
That's a really good question. So haven't really tried doing more than, I mean, I've done individual pulses into Europe and individual pulses into East Asia, but only a single one. I actually never tried doing multiple pulses, and I mean, I can do it, so why not? That's a really good question.
Thank you.
Did any of the scenarios you tried actually reproduce the right side of that graph? The like the higher coverage in Europeans compared to East Asia?
No, I don't think so. I think that's where that's what we need to work on right now.
Do you think that there's something like there's a scenario that's missing, or is the solution to be found in the scenarios that's being considered?
Yep, I actually not quite sure how to answer that.
No, it's a really interesting question. I'm just curious.
Yeah, no, and that is the side of that pattern that we're not quite seeing. I think we see it a little bit when we have both dilution and two pulses of introgression, but I'm not really quite sure. And it could be that I just need to adjust the second pulse of introgression or add a second pulse of introgression into Europe. But yeah, I'm not really quite sure. I mean, do you have any ideas?
No, I can't say I do. Yeah, I just wondered if you had any thoughts on that.
No, but thanks. It's still very much a puzzle.
Thanks.
Any final questions? All right. Well, thanks again for presenting for us, Fernando, and good to see you again, even though we'll be Zooming a lot in the future anyway.
Great talk.
Thanks.
Thank
Thanks,
you.
everyone.
Yep. So everyone have a good evening.
Yep. Bye.
Bye, Fernando.
Bye, Mara.